في ناس طبعا.
طيب يعني اول ما يعني انا ابدا على طول ولا حضرتك هتقول؟
هقول لحضرتك حاجه دلوقتي. طبعا اتفضل حضرتك، افتح المايك واشتغل.
بسم الله الرحمن الرحيم. اهلا بحضراتكم جميعا، وزي ما بيقولوها بالاردو عميديد، او بيقولوا برضه صبح بخير، ودول كلمتين the two words that I have recently learned from the Urdu language. So let's move on and keep the Urdu lessons for later. Hello everyone. For those who don't know me, I am Amr Abdu, professor in the Department of Microbiology and Immunology in the National Research Center. As we have today is a very special occasion of the Mother's Day, which happened also to be my birthday. So I'm off the hook, and when it gets to the Mother's Day gift. But I wish happy Mother's Day for all mothers joining our webinar today. This event was supposed to be in person, but we had to switch it to an online meeting due to the unprecedented situation of COVID-19, the global pandemic. And in this context, I would like to thank our guest today, Dr. Khan, for his patience and flexibility as he agreed to get online, although he was very excited about visiting Egypt. I hope soon this situation will change, and there's no need to wear masks, or there's no need for practicing social distancing, and we can meet him in Egypt soon. Before we start, I would like to thank Professor Mohammed Mahmoud Hashim, the president of the National Research Center, and Professor Mamdouh Muawad, the vice president for research affairs. I would like also to thank STDF for supporting this event, as they have different programs to support scientific events, which is very encouraging and helps in the dissemination of scientific information through the scientific community. The Department of Microbiology and Immunology represents a dynamic and exciting blend of disciplines. Our research and training are largely focused in the areas of immunology, bacteriology, virology, and mycology. And we also offer very comprehensive set of courses, seminars, and training opportunities for undergraduate students enrolled in the Faculty of Science and the Faculty of Veterinary Medicine in different universities around Egypt. We are also devoting a great part or significant part of our research activities to study the beneficial microorganisms. In this context, we have many running projects around this topic, and we have also made some successful seminars, including the 2018 seminar on the bacterial or the beneficial bacteria, and we were pleased to have Professor Bunia from the University of Bordeaux in USA as our keynote speaker, and he delivered two important lectures by this time. In 2019, we were also pleased to have him again in NRC to discuss how the beneficial bacteria can be engineered to stop the colonization of pathogenic bacteria in the intestine. We are now planning also for similar seminars and meetings to grab the attention of the scientific community and to discuss the ignored beneficial effects of viruses. And today's webinar, we will be discussing and unmasking the beneficial uses of fungi. The idea behind our interest in the beneficial microorganisms is that they are living in all kinds of environments, facing all kinds of harsh conditions and all kinds of enemies for millions of years. They have developed the power and the ability to produce many or certain metabolites to overcome these harsh conditions and to fight their enemies. As we are today facing life-threatening problems like drug resistance, the re-emerging infectious diseases and pandemics. Today, more than ever, we need to rediscover the power of these beneficial microorganisms, and to overcome these problems, we need to employ all the modern tools, techniques, and information to improve our ability to learn, to understand, and to apply what we have learned to satisfy our needs. I'm going to share just a few slides to highlight the uniqueness of and the importance of microbial products. As we see here in the process of drug discovery, it's a very long and very tedious process. It takes up to 15 years to develop the drugs from the laboratory to get to the market. And if you look here, it's like when you start with 10,000 compounds out of the laboratory, it takes 15 years and you only can get one compound approved by the FDA and gets to the market. At the same time, some of these drugs sometimes just get removed or get banned or excluded from the market due to their side effects. The reason is these compounds actually didn't get validated correctly before it gets to the market. So the main problem lies in the process of validation of the drug itself. Here we show some examples of landmark medicines produced by microorganisms. These products do not only have potent therapeutic activity, but also they have the desirable pharmacokinetics properties required to get to the clinical development, as they have been already validated through long history of repeated interaction with other biological molecules. These drugs are just few of the many microbial natural products that reached the market without any modification. As you know, if we go back to this slide, the most time consuming is the process of the preclinical and the clinical studies, when you need to do your experiment, both in vitro and in vivo experiments, and then you get your results, you go back to your chemical compound or your invention in the lab and try to modulate it, to change it, to overcome the problem of side effect or toxicity, and then you go back and try it again. But with these compounds, as you see, they all got to the market without any modification. So this actually shows the power and the ability of microorganisms to produce drug-like small molecules. This slide just shows the different applications of fungi and their products in industry, including the food, textile, animal feed, and biofuel. And this slide shows their application in agriculture, including their growth promotion ability, and they are used also as biofertilizers and biopesticides. I will stop here. I will stop here now, and it's my pleasure to introduce Dr. Khan. Dr. Khan is our guest speaker for today. He is assistant professor in the Center of Biotechnology and Microbiology, University of Peshawar, Pakistan. And he has basic degree in organic chemistry and master's degree in biotechnology and microbiology from the University of Peshawar. His PhD is hybrid from both the University of Peshawar and the University of Bristol in UK. He has also done his first postdoc in the Chinese Academy of Agricultural Sciences in Beijing, China. Finally, he is currently doing his second postdoc in genomics using next generation sequencing as a cutting edge technology. I will now hand it quickly to Dr. Khan without any further delay. Please join me to welcome Dr. Khan. Dr. Khan, please.
Thank you very much, Professor Amar. It was really an occasion for me, nice occasion for me to be here in this seminar, in this conference. I'm quite delighted to be a part of the coming and future programs. I'm thankful to all the faculty associated with your Department of Microbiology and Immunology. To be very honest, when you contacted me back in 2019, I was really very much excited to join you at your doorstep at your department. And I was curious to visit your institute personally, physically, and even to have had a chance to move around Egypt and to see some of the famous places as well. But as we all know, due to pandemic, that has not been happened that I have been to Egypt. But anyways, this is once again, I would say, your great efforts that you have just arranged this webinar. And we are all together speaking toward the same goal, same objective. Like how can we produce outcome out of the fungi, which is really very beneficial in some perspectives. I'm thankful to you, your seniors. I am sorry if I can't remember their names, but I extend my sincere thanks for allowing me to speak on this platform. Professor Amar, how can I share my slides?
Yes, you can share it now. You can just, did you see this small icon little there in your screen? It says share screen.
Okay, share.
Yes.
No share.
Yes.
Let me get into no share. Yes.
Great.
From here.
Yeah, you are in now.
Okay, that's okay. Is everything all right? Can you see my screen now?
Yes, we do.
Okay, thank you very much.
Sure.
So that was like the title of Unmasking the Beneficial Aspects of the Inconspicuous Organism, the fungi. Here on the screen, we can see right from the spore relation to different type of, and I have just given examples, provided examples of mushrooms, because this is like one of a famous saying in UK when you invite some of the prestigious person, you have to offer them with mushrooms. So as Professor Amar has just introduced me, I am Abid Ali Khan. My PhD is from Pakistan in UK. Then I earned postdoc from the Chinese Academy of Agricultural Sciences, and now I am working for earning a second postdoc in the cutting edge area of genomics. I would just start a quick start with the introduction. I used to say like the living organisms, which can exist anywhere in the universe. When I used to say this statement, what does it mean? It means exactly the same. I'm just talking about fungi. Now, if we can just see, I left like the industries, and I have for the first time used a term, which I have just used in one of my review, that fungi is a chemical industry, and I believe like fungi is a chemical industry, which is used for the production of biologically active secondary metabolites. Whenever we talk about biologically active secondary metabolites, So they are producing two different type of metabolites. The one that could be considered or developed into drug, and the second you can see at the bottom here would be the toxins. And both the metabolite, either if we can just drag it toward the drugs or either the toxins, they are biologically active secondary metabolite. Because from one side, we will look for the human's health. To improve health, we can lead to these type of beneficial metabolites. And for example, if we turn down toward agriculture, and there are some sort of toxic organisms, toxic microorganism, fungi, and we want to deal to stop producing toxin, there is even important to understand their secondary biologically active secondary metabolites. There are approximately 1.5 million species on this planet, and they are used as a source of food, particularly mushroom, which I have just started with the first slide, mushrooms. They produce secondary metabolite, which I have mentioned on a previous slides, like they could be processed to the drug formation, or either there could be some sort of toxin. The best example of the secondary metabolite to all the world is a penicillin produced by Penicillium notatum. As they produce bioactive metabolite, now it comes to researchers like you and me and the rest of the people in this area, where they are going to take it toward antimicrobial basis, if they are going to take it toward agriculture or either toward food. So that is totally up to the people. They want to play with the fungus. How it is on a screen, Fleming, Chen, and Frey. These were the three important guys who worked hard and provided penicillin, the one we are using as a marker that can inhibit almost all type of bacteria and that can kill. And that was like one of the best gift, I think I would say, after World War was available people, and it cures, it saves the life of the people around the world. Here you can see a map.
Dr. Khan, I'm sorry, it seems like Dr. Khan is like facing some technical problems with the internet. I'm trying to contact him to reconnect again. Okay, I think he's reconnecting now. Sorry for this inconvenience.
Sorry if you can hear me. I'm really sorry. There's some problem, some issue with the internet, but I'm committed that we can do it, inshallah, till the end.
Inshallah.
So Can you see my screen?
No,
Or
not really,
should
but
I share the screen again? Am I connected?
no, you're connected, right? But your screen is not shared yet. But I believe if you have problem with connecting your slides, I mean your presentation, like sharing it, so just maybe you would try, just go ahead and give your presentation until you are okay with sharing your slide.
It's okay. It's okay now.
Sure.
It's sharing now.
Sure.
Yeah, that's fine. So here you can see it seems like ambiguity, but this diagram shows the potential use of fungi in biotechnology. The cycle starts here with the basic biodiversity research, which in turn leads to culture placed in the central culture collection areas. The culture are then used for applied research, which in turn leads to product. For example, if we can just look into the diagram, sky color here, a novel compound used as biofertilizer and biocontrol, etc. And they are eventually lead to industries. Here you can see industries and the welfare of human being. The second area, you can see the light purple color. Use of fungi in agriculture perspective. Gene editing technology. Again, the gene editing technology for the welfare of human being. For example, if there is a fungus which is going to produce some toxic metabolite or materials, so gene cutting technology, gene technology we can work with that and can cut that gene, stop that gene from producing the toxic metabolite. The third portion here, you can see the cream type of color use of fungi in farming and food. The best example or the mushroom. This is just like a glimpse of fungal application for the welfare of human being. I just don't want to go into detail because I saw this slide during the presentation of Professor Amar. So, but still you can see like whenever we want to drag fungi, we can use it into multi different directions. This is a roadmap, and I think the first slide was reflected the same information like product development. It was even included in one of your first slides. So that's why I'm going to jump to another slide. Here, there is a selection of the target species. Now, question comes like if we can use fungus into various area or either into different direction, then comes the selection of the targeted species. We particularly do preliminary bioassay testing. And the following the bioassay testing, we can select either which fungus is beneficial or which fungus should we start playing with. Here you can see like two examples we primarily use in our research work, Aspergillus species and Cladosporium species. So we normally use these two species for the production of secondary metabolites. Post selection of the species, what do? We normally isolate and preserve that particular fungi. We then use isolation of the secondary metabolite using high throughput HPLC and the characterization of biologically active secondary metabolite using NMR technology. I would also like to focus somewhere here, like once we understand the nature of a specific fungus, through antibiosis or preliminary biosis screening, we use fermentation processes and then HPLC for isolation of a specific or targeted secondary metabolite. Then once we get out their secondary metabolite from the bulk of metabolites, we need another high throughput technology of NMR to reveal the mystery of the structure, to characterize the structure of that isolated secondary metabolite. And then it was like to understand the biosynthetic pathways of the active secondary metabolites by feeding labeled acetate. So this is like one of the chemistry perspective. And as you have just mentioned earlier in my introduction, like I am recently working in a cutting edge area of genomics. So we can even play with microorganisms other way around. And we can have a look on that as well. So while playing with fungi, we definitely need culture media because this is important to understand for those who are new to this area. Like wherever, what I mentioned earlier, like fungi can grow anywhere in the universe. Even they grow on the human's nails. Therefore, the environment outside of the laboratory is totally different. That is a natural environment. While environment in the lab is like a artificial environment. Therefore, we need to know and use different culture media. The media you can see on the screen. These are the different culture media, and this is not limited. The growth of fungus is not limited to these medias because we will still play and modify, optimize the condition of the nutrients as well as the time span for the growth of fungus or fungi. How to play with fungi in the laboratory? The first everything is like methodology, and I usually divide methodology. This is, to me, this is very much important for the new people to understand once they get a microorganism, any microorganism, despite of the fungus, and take it to laboratory, they need to know how and what to do with those microorganisms. So the methodology is phase one, I divided and into phase two. In phase one, I particular, this is my scheme of study. Like I go for the isolation and preservation, isolation from the mixed culture. Then the next step is to do the media optimization because what I mentioned earlier, outside there is a different environment in laboratory condition, there is a different environment. Then once we optimize the condition, for the production of greater number of secondary metabolite, we start doing preliminary antimicrobial testing to see if there are any potential molecules within the extract that they can inhibit the microorganisms, the pathogenic bacteria. Then extraction of metabolite and bioassay screening. In bioassay screening, we target bacteria. We can also target fungi, the pathogenic fungi. Then we straight away, if for example, there we got some sort of molecule within the extract that they can inhibit bacteria, as well as they can if they are not cytotoxic. I used to give example to my students, like if you can just take potassium cyanide, so that can kill everything, but eventually they can kill you. So you cannot recognize potassium cyanide as one of the best antibacterial or antifungal compound, or either it should not be used as antibiotic or antifungal drug. This is why after doing or conducting antibacterial and antifungal potentials, we straight away go and check the cytotoxicity if the extract is safe for the human's life. And then phytotoxic activity, like the extract is even safe to the plants, the plants kingdoms. Then comes to methodology phase two. And phase two, based on phase one, once it has been calculated that the fungus producing safe metabolite, we are going to isolate the metabolite, potential metabolite. Here you can see on a screen the column. We can even use column chromatography to isolate metabolite, or either we can use LCMS and HPLC, particularly preparative HPLC. We can day in and day out we use HPLC, and two type of HPLC. The one is analytical HPLC and the one is preparative HPLC. The preparative one is used for the isolation of potential metabolite. Then once we isolate a secondary metabolite, then the next step is to elucidate the structure. For elucidating the structure, we use 1D NMR and 2D NMR technologies. I'm not sure if your school, the people at your school, at your department of microbiology would use NMR or not, but still I would like to give you some sort of a bit information about the NMR. Like a proton NMR, a carbon 13 NMR is under 1D, and under 2D, just and depth. These are some of the 1D and 2D NMR technologies which we can do these experiments for revealing the structure or of the structure. I would use it like a flow sheet for purification of metabolite. The first ever is to grow fungus in the laboratory condition for the production of metabolite. You can see here, that one. The next is like bioreactor scale, and the third is like industrial scale production of the secondary metabolite of the potential or beneficial fungus. Here you can see this is an HPLC, which is, you can see on a screen a chromatogram. Is not clear, but does not matter. It just to give you an information. This is like a machine we can use for the, after doing these steps, these three steps, we utilize or use HPLC. The screen you can see chromatogram. Now I would say like most of the HPLCs are UV equipped or maybe diode array equipped. But I would suggest if those who are primarily interested in isolating potential metabolite using HPLC need to use mass equipped HPLC. Why mass equipped HPLC? Because mass equipped HPLC, every individual peak can provide an exact information of the mass, it ES positive and ES negative mood, and we can get an idea. Okay, let me just have a sip of tea. Thank you. Sorry. So that can give a clear idea if we can target any specific peak. Here you can see, let me just, here you can see different peaks. So if the HPLC will be mass equipped, we can propose like this this could be like a particular molecule, is not, it should not be a garbage. I would like to provide you with some information, like what I mentioned, what we do, right, starting from the antibacterial testing. The first one, optimization of the media. So normally, in our case, Aspergillus carbonis, the CGBS, CSB was one of the media, the best media for its production. Rhizi, the second media, you can say that was like CGSB, the same media for both the Aspergillus species, and for the Cladosporium CYB, and for Cladosporium caruni, that was like CYB. So there were like two medias. CGSB is good in our lab for the growth and production of metabolite for aspergillus species, and the CYB is for the production of metabolite for cladosporium species. Once we optimize media, we straight away, what I mentioned earlier, we straight away go toward the antibacterial activity, and you can see inhibition of the different pathogenic bacteria on crude extract. Here you can see different concentration of the crude extract, and here at this side you can see the pathogenic bacteria and the inhibition, percent inhibition as compared to the standard one. Say this side is like the inhibition of various pathogenic fungus strains. And that is like the cytotoxicity and phytotoxicity. So collectively, these 5 experiments, then we can get an idea either the fungus is good enough for us, or either it should be thrown back, or either should be handed over to someone he would like to reveal the toxicity of the fungus. That's why we select the fungus for our future activities. Here you can see some of the potentials or some of the routes which can lead toward the industrial production of antibiotic. Begins with screening for antibiotic producers. Now, how to understand which one is a producer? For knowing the nature to be producer of antibiotic, we should screen the fungus, or maybe sometime other microorganism lead toward the isolation after incubation isolation, and then you can see here their antibiosis, like to put in them together to see which fungus or either which bacteria can inhibit the other one. This phenomena is known as antibiosis, and based on antibiosis, we can screen out which organism is worth for us. The new producer are identified. Verification of chemical analysis of the entire microbial agent or performed. If the new antibiotic is biologically active in view, the industrial microbiologist may generate and modify up to their requirement or up to their desire. This is like one of the other perspective of our work that we can use indoor experiment, and this is like one of the streptomyces in KZ259. We have used their extract 100 times diluted. 100x means like 100 times diluted against the tomato plant and tomato roots as well as pepper plants. And there we got a significant increase in the biomass and even roots of tomato as well as the pepper plant. So after indoor experiment, we took those into the field experiment. Field most significant. Here we are using extract. We have never been isolation of metabolite with the control. The plant height was increased by 11.8%. The stem diameter increased down with 8%. And the same potential were observed against the pepper. You can see the conducted
معلش انا اسف جدا يا جماعه، واضح ان عنده مشكله في الانترنت، بس يعني هو بيقدر ان هو يدخل تاني، فهنستناه بس كده دقيقتين ان شاء الله يقدر يدخل تاني.
Okay, thank you very much for the patience. I'm really sorry for the continuous interruption. I'm really sorry.
That's fine. There's no guarantee that the internet is. Oh, yeah.
Yes.
Okay.
That's the biggest problem.
Yeah, I know. That's fine. Don't worry.
Okay, thank you very much. You are really a very kind-hearted person. I really appreciate that. Thank you very much.
You're pretty welcome.
Oh, thank you very much. Okay, so the same with the paper plant. Like the height were increased by 13.5%. The diameter 8 point something percent in the crown was increased by 16%. The maximum diameter of the fruit was increased by 13.27%. Here, so far, we are still using the extract and never use a pure isolated secondary metabolite. Here you can see isolated from flavors. In a sketch, you can see aspergillus flavors, then fermentation, extraction, LCMS, NMR, and again, and finally, we have got a molecule. You know, it's really very challenging to see a molecule somewhere, or either somebody just provide you with a structure, a complicated structure of a molecule. Particularly is my idea to give you some sort of information that we mentioned earlier. Here you can see the proton NMR. The proton NMR is one of the basic and essential experiment for the elucidation of the structure. I have just provided proton, carbon-13, quotient, HSQC of ethyl acetate, the very simple, just to give you a basic information. Like the proton NMR can gives us an information about the number of protons in a molecule. And I use a word, type of proton molecules. Sometimes my student asks question like proton or all the same throughout the universe, then why do we use the word type of proton? Type of proton means like when the protons are to be recognized based on their neighborhood. Like here you can see these three, the blue color, the two red, and the two green colors. These are the same hydrogens, but based on their environment, we can classify them as different type of protons, and they can give different peaks, and those peaks can provide us two type of information. Number one, the type of protons, and the number of protons means the type of hydrogen and the type of and the number of hydrogen. So from hydrogen proton NMR, we just calculate or this information to have how much of hydrogen in the molecule. Next, carbon-13 NMR. The carbon-13 NMR gives us information like how many number of carbon are there in a molecule. Like in case of, for example, 1, 2, 3, 4, ethyl acetate, there are 4 carbons. So the carbon NMR for ethyl acetate will give us 4 peaks, or you can see 4 lines, and that would be the identification, or it's like a prediction that there are 4 carbons in a molecule. Then comes to HSQC. This is really a very, very interesting, a very informative experiment, because so far we know how many number of protons and how many number of carbon are there. The HSQC gives us a clear information, like which hydrogen or connected to which carbon, which hydrogen or how many hydrogens are connected to which carbon. Like for example, you can see here that these three hydrogens are connected to this carbon. So this information, the connectivity of these hydrogen to this carbon is same like the hydrogens are carbon formation by HSQC. This is said to be fragments or fragmentation. We deduce various fragments using HSQC. Now the next step is HMBC. Then HMBC provides information the molecule or the structure of the molecule. But again, I say, like once we construct the structure, this is still not even enough unless we do not conduct the COSY. The COSY experiment then provide us with the information of neighborhood protons. If the protons are connected to different carbons, but they exist I think I have given a shot over techniques like RMR or carbon 13 NMR, HSQC, and HMBC and COSY. These are the essential techniques for the structure elucidation. Over to the latest technology, the correct analysis with the secondary metabolites. I'd like you an overview like NGS technology is the only technology they have just given us information of science or revealed the information, and now we have the information of fingers. Understand what is the platform of NGS and how does NGS work for us. So if you can just see, let me just make bigger. If you can just see is a sample or a genome. Here you can see the DNA, the genome. Every living organism have the same A, G, C. Then what we want to do is actually fragmentation. We cut all those genomes into pieces, and then the next is like we provide adapters. So that step is said to be adapter ligation, and that step is actually for the identification of a specific fragment which could be overlapped after sequencing. Then doing these three steps, we straight away come here. This is like a PCR amplification, and based on the PCR amplification, the phenomena is exactly the same PCR amplification. But here you can see here you can see different colors. The different color represents like different adapters attached to different fragments. So these are the adapter or indices which can be later on used to break one fragment. I will give another. I can give you an overview. Here you can see this is like a pair-end sequencing. The phenomenon is a PCR. The adapter used the primer. So at one end, you can see the adapter, and this like flow cell.
the rectangular you can see is like the flow cell. So on a flow cell, there are complementary adapters. So the adapter to the genome, they are complementary to the adapter on the flow cell, and based on the complementary, they ligate it to each other, and then you can see three different parts. The one is like pyro sequencing, Roche sequencing. The other is the by ligation, and the third is sequencing by. What I mentioned is NGS, Illumina NGS. We are primarily dealing with MySeq, so they work on the phenomena sequencing by synthesis. Okay, this slide is a blurred slide. I didn't like this slide, but unfortunately, what I mentioned, like I have been through a lot of emergencies for the last couple of days, so I didn't find another clear slide. But here you can see the sample pre-processing the DNA. First, you need to bring out the DNA, and then the DNA is a library processing to fragmentation, attachment of the adapters. Then the adapters, the library, this is called a library preparation. The library is going to be pooled and put in the cartridge to the sequence, and then we can have a sequence like this. And what can we see here is basically the overlapping, and from these overlapping we can deduce the exact sequence like you can see here. Are the bioinformatics tools which can do these type of bioinformatics tools which can align the overlap, and after overlapping there are like statistics working cameras and algorithms within the bioinformatics tools and get a clear sequence. This is one of the examples of our 59. We do sequencing. The gene length distribution, we can get a clear-cut information about the genes. Say, for example, we are interested in a particular gene, and we have screened a sequence clearly, Professor. Maybe is again, there seems a problem. Seems a problem with the signals. Because yes, I think we should know. But really, okay. So please let me wait. Slide. Sure. What is you or signal this slide? Okay. Please provide me okay. So I am
right slide.
Yes, I've sent you the slide that we can see over the screen right now. So, but you're still cutting out. I don't know. There's major problem with the internet. So
Okay.
So I would just suggest if you can.
It's okay.
Sure, sure. أنا آسف جدا يا جماعة وبشكركم على أنا شخصيا عندي مشكلة في النت حتى التكنيشن كان بيحاول يعني يحلها لي النهارده وشكله ما قدرش طبعا فأنا حتى بشتغل بالهوت سبوت بتاع الموبايل بتاعي ف دي أزمة خصوصا لو حد بيتكلم من بره مصر فهننتظر شوية برضو تاني إن شاء الله والله المستعان إن شاء الله يقدر يخش تاني. انا بقترح عليه ان هو لو يقدر يعني يتكلم من غير ما يعمل شيرنج السلايدز بتاعته واضح ان الشيرنج بتاع السلايدز عامل مشكله في الانترنت نشوف هيقدر يعمل كده ولا لا ولما يثبت مع الانترنت ممكن يحاول يعمل شيرنج تاني للسلايدز.
Okay, I'm sorry for inconvenience. Can you hear me?
Yes. Yes, we do now.
Okay.
Sure.
Oh, thank you very much. Like our platform, they do sequence synthesis. So after synthesis or sequencing, we have like a snapshot, the screen, oh shit. Sample pre-processing, and then library preparation, and after library preparation, actual sequencing by using MySeq machine. So after sequencing, we are getting a lot of data in the form of FASTQ files.
and there is actually a puzzle. Therefore, bioinformatics tools are required to deal with those puzzles and to get a proper or specific sequence like the one. I'm not sure if you can see the screen or if you have shared the screen. Then by sequencing, it gives all the information about all type of genes. Now, if we are interested, for example, in any specific gene or even a cluster of genes in terms of fungal secondary metabolites, we can send the genome of fungus for sequencing. And these days, the sequencing cost has exponentially decreased. Thus, we can get the profile of all the genes of any particular microorganisms. Let's say we are interested in gene A, and that gene A is present within the genome of a specific fungus. We need to deal with that fungus. Otherwise, we should not deal with the fungus particularly. Like here you can see, I'm not sure if you can see the analysis of the whole genome and the information of all the genes of NKZ259 strains. So there were like 36 gene cluster secondary screen sharing. Okay, please. Secondary metabolic gene cluster of 36, while 29 were involved in regulation of indole synthesis. Keeping in view all the genomic information, it comes to the role of any specific or particular gene, like here I have mentioned WYSP2 in voyeunicine production and morphological development. The gene replacement, sorry, the gene represented, here you can see, no, the WYSP2 in albulus CK15. The role of WYSP2 gene in the
وانا عملت شيرنج للسكرين يا جماعه مكانه، هو كان باعت لي المحاضره وانا عملت شيرنج للسكرين، بس ننتظره هو يدخل تاني.
Okay, so here you can see like gene clusters. This is like gene map, and here you can see gene distribution, even including the number of genes, type of genes, gene cluster, and gene length. Now, once we get the whole genome sequencing of any particular organism, we can get an idea which gene or gene cluster we are interested in. Therefore, we do not need to go back and isolate any fungus or any other microorganism without any information to take them into laboratory condition, provide them with an environment, optimization of media, all those type of steps. We can eliminate all those steps. Just a sequencing information is enough to know if there is any gene of interest or not. Next, please. If you can just get into next slide. Next slide. Uh, uh, sorry, bad internet disrupted and make, I think, little bit terrible. But anyways, we are just 2 slides away. Let me just conclude in this particular way. Like once we have the information of all the genes or a cluster of genes, the one we are interested, we will select that gene, and then we can see and work with that specific gene. Like the second last slide, yes, where I have just provided a snapshot from our. Oh my goodness. Actually, once we determine there is any gene of interest, we targeted YSP2 gene, which was responsible for the vaccine synthesis. Yes, and morphological development. Thus, the disruption was carried out to our plasmid, and thus we were able to play to see if their gene is responsible, because from the genomic analysis we were concluded like the gene is that specific gene is present within the genome of Albulus. Okay, so yeah, I was just talking about this slide. Like we were interested in this gene. We got the information about this gene from WYSP2 from the genome, the sequencing data, and then we work with knockout experiment to confirm if there is a role of this gene. This could be like one of the best approach to have a and then we can correlate the chemical role of metabolite with the specific gene. So for the future, you can please get to the next slide. Oh, okay, Professor. I think this is not the last slide, the future recommendation. The one I have modified is like the way we have the cutting edge technology, high throughput sequencing, next generation sequencing technology. We need to go and put our hands into metagenomics. any type of microorganism we want to deal, we can reveal the mystery by knowing the metagenomic approaches. So the future recommendation is about the metagenomic approaches. Thank you very much. I will just put forward to the acknowledgement. Like, I acknowledge University of Peshawar, University of Bristol, CAS, the Higher Education Commission of Pakistan, and my current institute where I work. Thank you very much for the patience, and I'm really sorry for the continuous disruption during the session.
Thanks, Dr. Khan. On behalf of everyone here, I thank you so much for your valuable presentation. I particularly enjoyed your way, when you have brought down together all the steps from the isolation to purification and toxicity check for the secondary metabolites. I was also excited with, to know about, using the fermentation process to promote the growth of plants. And I'm pretty sure that some, especially of the companies of the, the private sectors would love to know that. So now, thanks again. And if there's any question, we'll try to answer it, please.
Yeah, sure. If there is any question from the audience, I'm here to answer the questions. And if you would have just given me this opportunity to ask question from you, so I have no question. I am thankful to you and your seniors, your junior, all those who participated and provided their valuable time.
Sure. I actually want also to thank, the conference unit, especially Dr. Hiba Rifai and everybody working in this unit. Actually, they are doing great job switching from, the in-person meetings to the online seminars, and it has been great job. So استأذنك يا شريف لو في حد من الحضور عايز يسأل سؤال ولا حاجة تساعدنا في الموضوع ده.
Management sequencing caro, Bristol caro. بس almost ختم شو.
Would you please identify yourself again? Just introduce yourself to the audience, please. Make it more...
Me?
No, I'm sorry. There was somebody asking a question and...
Okay.
شريف احنا معانا حد بيسأل حاجة مش كده؟
كم دور بدون التركيبان دول
Okay, Dr. Khan, I think we'll stop here, and we thank you again for your presentation, and I hope the situation will get much more better soon. And we had some like internet cutting off in some of the slides, so I wish we'll meet in person soon in Egypt. So thanks a lot, and thanks for everybody.
Yeah, thank you very much. It was really an honor for me. I really respect you. I really respect the audience for listening to me during the disruption and interruption of the internet. And thank you very much for the opportunity that I have just been a part of this seminar. Thank you very much.
Thanks.
And see you.
See you. Bye.
Thank you very much.